After we've seen these beautiful presentations with photography and singing, I'm going to bring us in a decidedly different direction a little bit by talking about the way that we study how our brain is capable of making these types of things, of doing these complex tasks. What I'm going to talk about is a story of the leech and the lens, and it's the way that we study neural complexity and the way we study nervous systems. So I'm a physicist at the University of California, San Diego, and inherent to being a physicist, my sort of favorite philosophical tool to use is called reductionism. Now, this is the idea that in order to understand something, what I like to do is to break it down into its component parts. Now, what this is to say is that if I've got a difficult phenomena that I want to understand, I could take the fundamental components that make it up, study those components, study their interactions, and then I can understand the phenomena in a very deep way. Now, to give you an example of this, your wristwatch is a perfect case. You could start out by taking your wristwatch apart with a screwdriver, which I've done, by the way, and the trick to doing that is to put it back together without any spare parts left over at the end. But once you've done that, you'd see gears, springs, axles, wheels, and those are all mechanisms that are understood by basic mechanics. Now, if I were to measure each of these individual gears and where it's located in the watch, I can start to draw conclusions and make statements about their function in the watch. So as an example, I could be looking at the gears and I could say, okay, this set of gears is going to rotate such that the axle is going to turn at 1 revolution per hour. That has to be the hour hand. And understanding the parts and interactions in this way tell me those interactions, like the hour hand, that actually make a watch a watch. And reductionism works as a nice tool for the physical sciences. But I'm interested in studying biology, and that's where things start to get a bit tricky. So everybody here has one of these, right? So our brains are, now Joe from Gunnar Optics and I are probably going to get into an argument about this later, because I think the brain is actually the most complex part of the human body versus the eye, but it's sort of semantics. Our brains are capable of doing some truly amazing tasks. As an example, introspection, the idea of looking in on oneself. Introspection itself recursively leads us to want to understand introspection. So that's just a taste of how complicated these tasks can actually get. And that's where the problems come in. Complex systems like the brain, they're really hard to tease apart. If you start dismantling the brain, you're going to break it without revealing any additional structure or function. So a brain taken out of the body and dissected, it doesn't work. Now, that isn't to say that we don't learn anything by dissecting brains, which neuroscientists tend to do. We've learned that there are centers for vision, centers for touch, centers for hearing. We've learned that there are cells that are called neurons, which interact and send signals, and that's been known for hundreds of years. However, unlike the watch, where the second-hand mechanism could be taken apart and understood on its own, and built on its own, in fact, a dissected brain is fundamentally different from an intact brain. The level of interconnectedness and the density of interactions makes the brain fundamentally greater than the sum of its parts. And for that kind of situation, reductionism fails. To understand the brain, we have to understand it in situ as it functions. We have to look at it working. And to understand it deeply, we need to know the microdynamics that are only observable by taking it apart. So what you have is a catch-22. If you take apart a complex neural system, it's no longer a complex neural system. It doesn't work. But the only way to understand that type of system is to look at the microdynamics, and we can only see that by taking it apart. So what do we do? So my answer, maybe? There we go. So my answer is that we should study the simplest animal that we can find with the most complex behaviors. Somewhere in the animal kingdom, there's a local minima for complexity with a local maxima of interesting behaviors. Now, I believe that intersection is Hirudo verbana. That's a medicinal leech, a European medicinal leech. Now, the leech is an invertebrate annelid worm. But it also happens to be a complex predator. I don't know if everybody knows this. I didn't know it before I started working on them, but this is a hunter. It eats blood, and in order to do that, it has to hunt down its food. And in order to do that, it uses only a small set of behaviors, a characteristic repertoire. So it's got local bending, which is where it bends its body to avoid a touch. It's got whole body shortening, which is where if you tap it in the front, it'll contract to avoid the stimulus. It's got swimming, crawling, feeding, and mating. So now Now, even better, every single leech is alike. They all develop in a characteristic way. All the behaviors are the same. Every time that a leech grows up, it's going to look the same. Their nervous system is laid out in the same way. And they're an invertebrate. They've got a soft body wall. So we can surgically look at the nervous system. Now, the thing is, it's still got about 10,000 neurons. So why am I saying that this is the minimally complex, maximally interesting animal? The answer to that is actually in the layout. Now, the leech has what's called a metameric mid-body nervous system. So all this means is that the leech is segmented. It's got one bundle of carefully organized neurons in each segment, and that's called a ganglion, and that's the word I'm going to use to call it for the rest of the talk. Now, each of these ganglia, especially the ones in the 21 that are in the middle, are essentially identical. And each ganglia is actually responsible for generating the behavioral output of the animal in that segment. So this means that the leech, although it's 10,000 neurons, is already a reduced problem. We're down really from about 10,000 neurons to about 400 neurons. And because each of those is paired, there are only about 200 unique neurons in the leech. And they're just repeated down the body. So we can sort of say that the fundamental unit that makes up a leech is this 200 cell unit. That's the gears that make up the leech. And because there are so few neurons, most of those neurons are unique, and when we stimulate them, they'll actually change the output. We can stimulate one neuron electrically, and it will change the behavioral output of the animal. But that's still not all. The other really huge advantage to the leech nervous system, and why I'm here talking about it today, is because the leech nervous system, unlike any mammalian system or any higher animal, can function when it's actually removed from the animal. If we remove the body wall, the skin, and the muscles, we're left with the nervous system, and you can see that in the bottom of the left slide. Now, we can then record from the nerves that would normally be talking to the muscles and listening to the skin. And we can observe signals that we know from experiment indicate fundamental behaviors that the leech would be exhibiting. For example, those patterns up there, which are electrical recordings, are known to represent swimming. When we see those patterns, we know that the leech is fictionally swimming. It's trying to generate the signals that if it were hooked up to a body wall, it would be swimming. So what this is saying is that the leech nervous system and brain actually works when it's outside of the animal, unlike any other higher animal. And that lets us understand things with an amazing level of detail. Because we can see all the active neurons in a ganglion while they're working, and because we can correlate that to the behavioral patterns, we can now start to make measurements of the microscopic dynamics of the nervous system. Now we do this by taking a leech ganglion, and there's actually a picture of it on the screen, and we stain it with a special type of fluorescent dye. Now, we then use a high-speed camera, to image the fluorescence. And we put little regions of interest around each of the neurons, and we can track the fluorescence for each particular neuron. And that actually tracks the activity. The fluorescence from these special dyes tracks what the neuron is doing and reports it optically to us. So I'm a physicist, and what we call this is a state measurement, and this is sort of the lesson for the evening, is that I'm going to teach you what this is called. A state measurement is the unique set of activities in every cell, in other words, the position each cell is in, and then we follow that as it evolves over time. It's the unique state of the animal, so we just call it a state measurement for simplicity. Now, the human equivalent of this would be like measuring the activity level of all 50 billion neurons that make up one of your hemispheres. It's a completely different problem scale. I mean, this is the difference between looking at a wristwatch versus an atomic clock. They're just entirely different problem scales. This is addressable. That one's not at the moment. Now, because we can see all these cells at once, and actually what you're seeing on the right is just a trace of all the different cells over time as they evolve. So because we can see all these at once, we actually see new things. A really good example is the following. We can see that right before the leech makes a decision to, say, swim or crawl, there's a certain subset of neurons that become active in a certain order. But this is actually happening before the decision, and we're seeing it optically, and then the behavioral output begins electrically signaling. So what that says is that subset of neurons that we can only see by seeing all of them at once is actually responsible for making the decision in the animal. Those neurons are deciding what the animal's going to do. So we're effectively reading its mind optically. Now, the analogy to this is that it's like owning a watch with a transparent front and back. You can sort of if you could watch the whole device in action, if you can see the watch and the inner workings as they move around, you could understand it just by looking at it, and that's sort of what we do here. And because we know the order of the activity and the strength of the activity, we can actually write it out as a circuit diagram like you would for an electrical system. Now, that's a huge difference, because now we're suddenly able to understand in detail what's going on in the decision-making process in a microscopic way, which has never been done before. But that's sort of not the entire thing. If we want to prove that these circuit diagrams that we're designing and discovering are correct and complete, we would want to imitate the signals that are coming into the ganglia in real time and show that if you issue these commands to the circuit will respond by making the following decision, and we can say, I'm going to command the leech to do a following behavior. We can't do that yet. We're still limited in most labs to driving a single neuron with a glass electrode stuck into it, which means that we can't imitate complex and dynamic activity patterns. In our lab, I'm actually working to solve this problem by activating neurons with laser light. So you can imagine a moving projection of points of light, just like a movie projection. And I could put a particular pattern onto the ganglion, and that would actually command the ganglion to begin crawling or to begin swimming. So these different targets, which is all done with light, they can simultaneously be done while we're measuring the responses. So this simultaneous measurement and control, it's like being able to control the speed of every single individual gear within a watch and being able to set them all in motion at particular speeds and then watch the response. It's a really fantastic way to actually understand how the circuits work, what their layout is, and how they respond to changes. So the idea that I really want everyone to take home from this is simply the following. If you want to understand complex systems, especially like our brains, we need to understand the basic circuits that allow any animal to have drive and behaviors. Now, in order to understand those basic circuits, we have to be able to observe them in action in a detailed way. And by using these advanced optical techniques that we're working on to study the nervous system of the leech, where we can measure and interact with a functioning and working nervous system, we can understand these fundamental circuits that generate the behavior. And once we understand those circuits, we know what we're looking for. And we can then take our knowledge and begin looking at the higher for the more complicated implementations that have to exist in higher animals like mammals, chimps, rats, people. And we have a starting point. And it makes the problem solvable, because 50 billion neurons interacting in a very complex way when we can't see inside them, we can't access them, is a very difficult problem to solve. So what I'd like to get across tonight is that you shouldn't forget the leech. The best way to understand ourselves is to learn everything we can about this interesting little invertebrate. Thank you.